Okay, so we've got a question here on plant and animal cells. It says draw one line from each cell part to the correct function of that part. So first of all, what does a cell membrane do? A cell membrane controls what goes in and out of the cell. So the answer here would be controls the movement of substance into and out of the cell. The mitochondria is where aerobic respiration occurs, and that's where most of the energy of a cell is released. And finally, we have the nucleus, which has all the DNA, and the DNA is what controls the activities of the cell. So you can draw a line there. Second part of the question, we've got a plant cell here, and it asks us two parts of the diagram that are not found in animal cells. So we've actually got three things that are not found in animal cells, and that would be the chloroplasts, the cell wall, and the permanent vacuole. The permanent vacuole is not labeled here, so we can just go with chloroplasts and the cell wall. So if you got this 4 marker in your exams, would you be able to get all the marks? So here it asks us to describe 4 differences between a bacteria cell and a plant cell. So the main difference between the two is one is prokaryotic and the other is eukaryotic. The main difference between the two is that a eukaryotic cell has all of its genetic materials in a nucleus, whereas in a prokaryotic cell, it isn't in the nucleus, it's just in the cytoplasm. So that's one of the differences that we can say. We can say plant cells have their DNA inside a nucleus. Another difference between the two is that bacteria cells are much smaller than plant cells. So plant cells have chloroplasts, whereas bacteria cells do not. So you can say plant cells have chloroplasts, but bacteria do not. Another thing that plant cells have that bacteria do not is a large permanent vacuole. Bacteria cells do not have those. So we can write down plants have a permanent vacuole. And there you got it. You've got 4 marks. All right, let's try this 5 marker on cells. So here we've got a question where it says draw one line from each cell structure to the type of cell where the structure is found. So the difference between prokaryotes and eukaryotes, remember, eukaryotes have a nucleus whereas prokaryotes don't, or their DNA is just floating around in the cytoplasm. So you can memorize that by remembering that eukaryotes have an EU in it, just like the word nucleus. So that reminds you that eukaryotes have a nucleus. For prokaryotic cells, the genetic material is not found in the nucleus. Instead, it's floating around in the cytoplasm or found in rings, and we call those plasmids. So that's the next answer. Plant cells are the only ones that have a permanent vacuole. So that's their store of sugars and salt solution. Now for question B, it says cells contain structures. These structures have different functions. Draw one line from each function to the correct structure. So which of these contains genetic material? So we've done that in the previous question. The nucleus contains genetic material, the DNA. What controls what enters and leaves the cell? That would be the cell membrane. And where does photosynthesis occur? Photosynthesis occurs in the chloroplast where there's chlorophyll present, which absorbs sunlight for it. Easy 5 marks. So the question is specifically talking about water. So root hair cells also take up mineral ions. Because they mention water here, we have to talk about the water in our answer. So the main thing to note from root hair cells is that they have these hairs that stick out, and that increases the surface area. So it has a large surface area. But this is a 2-mark question and it says explain. So why does it have a large surface area? So remember, it's talking about water here. So we need to talk about osmosis. Yes, it does also take up ions through active transport, but the question is specifically talking about water here. So we need to talk about osmosis. So the large surface area makes osmosis occur faster. And that's how you get 2 marks. So what kind of cell is this? The clue here is that it says it absorbs substances from the soil. So you find this in a plant and you can see that it has this long hair that sticks out. This is known as a root hair cell. 1 mark. Describe how the root hair cell is adapted to increase the absorption of substances from the soil. Well, it's got this long hair which increases its surface area. So that means it can absorb substances faster. So we can say it has a large surface area. Okay, so here we have a diagram of a sperm cell, and the question asks you to draw one line from each feature to show how the feature helps the sperm cell. So first we have it contains a nucleus. Why does it contain a nucleus? Because it needs to carry the chromosomes, the 23 that it has, from the sperm to the egg cell so that they can fuse to form a full set of 46 chromosomes. So the answer here would be to provide the chromosomes for fertilization. The second, it has a long tail. Why does it have a long tail? Because it has to swim to the egg. So the answer would be there. Right, what are the differences between the xylem and the phloem? So here we've got a 6 marker on that. So it clearly says compare the structure and function. So let's split up our answer into those two sections. So in terms of structure, the xylem is made up of dead cells, whereas the phloem is made up of living cells. And because the xylem is made up of dead cells, it needs to be strengthened, and it's done with a substance known as lignin. So xylem tissue is strengthened by lignin. Another difference that you can mention is how their end walls look like. So phloem tubes are made up of end walls with tiny pores in them, whereas xylem tubes don't have those end walls, they're completely hollow. So xylem is hollow, but the phloem has end walls with pores. Now let's talk about the differences in their function. They both transport things, but the xylem transports water and minerals, whereas the phloem transports sugars. So we can write that down as the points for the function. The xylem transports water and minerals, but the phloem transports sugars. So we're nearly there. So the next thing we can do is talk about the direction of the movement of the substances. So for the xylem, the substances, the water and minerals, only travel upwards, whereas for the phloem, the sugars can move both up and down. So xylem transports water upwards only, but phloem can transport sugars up and down. And finally, the phloem is involved in translocation, whereas the xylem is involved in transpiration. And for all of that, you get 6 marks. So here's a question on the required practical on microscopes. Here it asks us why it's easier to observe a cell using the lower power objective lens first. The reason why is because you want a big field of view before zooming into what it is that you're looking at. If you were to look at an image with a high magnification first, you'll find that you won't know where you are and you won't know what you're looking at. But if you start with a big field of view and slowly zoom into what it is you're looking at, it's much easier. So for this answer, to get 1 mark, you have to say it's the biggest field of view. So by having the biggest field of view, you can then find it much easier to focus on the cell. The second part asks us to focus the image, the objective lens should be moved away from the stage. Why should it not be moved towards the stage? So if you look at the diagram of the microscope, you can see that if it was to move closer to the stage, there's a chance that it could come in contact with the stage, and that could damage the lens. So to avoid that, you need to always make sure that you're moving away from the stage. So the answer for this one, to avoid damaging the lens. Now the final question is just a very simple calculation. We have an image of a slide and it says that it's viewed with a ×10 magnification with the objective lens. So remember, there's 2 lenses involved in a microscope, the objective lens and the eyepiece lens. So the objective lens has a magnification of ×10 and the total magnification of the whole microscope is ×50. So the question is asking for the power of the eyepiece lens. So what you need to do is multiply the magnification of the eyepiece lens with the magnification of the objective lens, and you should get the total magnification. So that would be 10 times the magnification of the eyepiece lens, and that should equal to 50. So we're trying to find out this number. So something times 10 is equal to 50, and the answer is 5. So if your eyepiece lens is ×5 magnification and you multiply that with ×10 for the objective lens, you should get a magnification of ×50. So the answer here is 5. So here we've got a question on the required practicals on microscopes. So what we have here are 2 images, and there's something wrong with both images. It says in the question that both images have cells which are not clear to see. And it's asking you, how should we adjust the microscope to see the cells more clearly? So each one has a different problem. So slide A, you can see that the cells are slightly out of focus, they're blurry, so it needs to be focused more. How do we focus it on a microscope? We need to use one of the focusing knobs. So the answer here is turn the focusing knob to focus on the cells. The second one, slide B, you can see the cells, but it's very zoomed out. We need to zoom in more into this picture. So how do we do that? Well, remember, we also have objective lenses. We have 3 objective lenses on a microscope. So what we can do is we can rotate the objective lens to a higher magnification to zoom in. That's how we get 2 marks here. You mentioned both of those points. So first point is to say, rotate the objective lens. And for the second mark, you say to a higher magnification. And that's it. Now, if you were to get a calculation on biology paper 1, it's most likely to be a microscope question like this one. So here we have a student measuring the length of a palisade cell. What we have is the cell image is 28 millimeters and the magnification of the microscope is 400. Now we need to calculate the real length using the equation for magnification. So what they've done here is they've actually rearranged the equation for us, which makes it a little bit easier. All we need to do is put the numbers in now. So the image length, as stated in the question, is 28 millimeters. So we do 28 millimeters divided by the magnification, which is 400. So 28 divided by 400. And if you put that in your calculator, you get an answer of 0.07 millimeters. The second part of the question asks us to convert the length from millimeters to micrometers. So here you need to remember the conversion. So to go from micrometers to millimeters, what you need to do is divide by 1,000. And to go from millimeters back to micrometers, you just need to multiply. This question is asking us to convert from millimeters to micrometers. So that's multiplying by 1,000. So what we need to do in our calculation is 0.07 multiplied by 1,000. And that gives us a final answer of 70. Do you know all 3 stages of the cell cycle? So here we have an exam question where we have all 3 stages on a pie chart. And it asks us to identify what each stage does. So stage 1, what happens? So remember, mitosis is when a cell divides into 2 new daughter cells. So what it needs to do before it divides is it needs to replicate everything inside of it. So that includes all of its subcellular structures like ribosomes and mitochondria, as well as replicating all the DNA in it. So here, what we would say for stage 1 is copies of the DNA are made. For stage 2, that's when mitosis happens. And that's actually in the pie chart. So the nucleus has all of the X-shaped chromosomes, then get pulled apart to opposite ends of the cell, and new nuclei are formed. So the nucleus basically divides. Once that's done, you've got 2 nuclei, you've got all your extra subcellular structures. Now the cell needs to divide. So that's the cytoplasm and the cell membrane. Both of those divide to form 2 new daughter cells. So that's stage 3. Okay, so here we've got 3 different diagrams of cells at different stages of the cell cycle. The first question says, which photograph in figure 1 shows a cell that's not going through mitosis? So in the first one, you can see it is going through mitosis because that's where the cytoplasm and the cell membrane is dividing. Image B, it is also going through mitosis because what's happening here, you can see the cell fibers pulling apart the X-shaped chromosomes to either side of the cells. So that means it is in the process of mitosis. The answer here is C, guys, because in C, it's just a complete cell. There's nothing going on. There's no cells dividing. There's no nuclei dividing. There's clearly a nuclear membrane here, so that hasn't disappeared. There's no free-floating chromosomes or anything like that. All the chromosomes are very much in the nucleus, so there's no mitosis occurring in that example there. So the answer here is C. Now for part B, it says describe what's happening in photo A. So I told you already, this is where the cytoplasm and the cell membrane of the cell is dividing. It's the last stage of the cell cycle. So it's 2 marks. So let's mention that for 1 mark. Cytoplasm and cell membrane is dividing. Now for the second mark, you need to say what happens at the end of it. What happens? The one cell is now turning into 2 daughter cells. And remember, those 2 daughter cells are identical. So we could say to form 2 identical daughter cells. That's how you get 2 marks there. Now, Part C of the question. We've got a table here. A student wants to find out more about the cell cycle. The student made a slide of an onion root tip. She counted the number of cells in each stage of the cell cycle, and the table below shows the results. So total, it says 36. She looked at 36 cells and she noted down at what stage they were at. So 20 of the cells weren't even dividing. They were just on their own. All right, so they weren't in the process of dividing. Then we've got the rest of the cells, which add up to 16. So 16 of the cells are in the stages of the cell cycle. So it says 9 of the 36 were in stage 1, then 4, then 2, and only 1 was in stage 4. And the question's asking each stage of the cell cycle takes different amount of time. Yeah, so that's true. Usually the first stage, where the DNA replicates and the mitochondria and the ribosomes double, that process usually takes much longer than the others. And it's saying which is the fastest in the cell cycles based on that table. So if she's seen 9 cells in stage 1, that means that's probably the one that takes the longest, because it's more likely that they're in that stage. So they've spent a longer time there. The one which takes the shortest time is the one which has the fewest number of cells in that stage. So if it takes a split second, then you're likely to miss it if you were to take a snapshot of the whole 36 cells, right? So the one that's probably the fastest is stage 4, because only 1 cell was caught at that stage. So the reason why you'd say only 1 cell is seen at this stage. Here's an exam question all about mitosis. So the question asks us to describe how mitosis produces 2 genetically identical cells. And it's actually 4 marks. So that means you need to be quite detailed here in your answer. So if we look at our stages for mitosis, you can see the first thing that happens is DNA replication. So here all your DNA is doubled and duplicated. So that's one point we can write down. The genetic material is duplicated. Now what happens next? You have these X-shaped chromosomes, and the X-shaped chromosomes are pulled apart to opposite ends of the cell. So you can see that process happening here. So you can say that as your second point. The chromosomes are pulled apart. Now for the third mark, what happens after they've been pulled apart? The new nuclei form, and the cytoplasm and the cell membrane now divide into 2. So that's shown in this stage. The cell membrane and the cytoplasm divides into 2 cells. You can also get marks to say that the nucleus divides, because remember there's 2 new nuclei that are formed on either side of the cells. Now we still need 1 more mark. Where do we get that 1 mark from? So you need to remember you have the 2 daughter cells, and the 2 daughter cells are identical to each other. So you can mention that at the end. So the question itself says, how does it produce 2 genetically identical cells? So you need to describe why the cells produced at the end are identical to each other. That's because the chromosomes inside of it are identical. So that's what the 4 marks is for. You could say the chromosomes in both cells are identical, and that gives you 4 marks. Which of these statements about stem cells is correct? So the first one, animal stem cells are found in meristem. Nope, that's not true. Animal stem cells divide by meiosis. No, they don't. They divide by mitosis. The only things that divide by meiosis are gametes. Meristem cells in plants can differentiate throughout the life of the plant. That is the correct one. They can differentiate at any point in their life. It's a bit different to human stem cells because they can only differentiate at a very early stage of the human life. But for plants, they can differentiate at any stage. Meristem cells in plants can only differentiate into one type of cell. No, they can't. Plant stem cells can differentiate into any type of cells. So the answer here is the third one. Okay, so we've got an easy definition here for what is a stem cell. So it's 2 marks. So you can't just say it's an unspecialized cell or undifferentiated cell. You have to say 2 different points. First mark, we can say that. We can say an unspecialized cell, but you have to say more now for the second mark. So what is it that we can say? What can an unspecialized cell do? It can differentiate into other types of cells. That's the definition of a stem cell. And that's how you get 2 marks there. All right, so here we have a question on stem cells. So it says scientists have developed patches of beating heart cells to repair damaged heart tissue. The patches are placed onto areas of the heart where cells have died. New cells grow to replace the dead cells. So they're applying these patches onto wherever the damaged area is. And what that does is it causes new cells to grow in that place. So basically repairing the heart tissue. The patches are made up of the person's own cells that are converted into stem cells. So explain why stem cells are used to make these patches. So remember what stem cells are. They're undifferentiated cells with no job. So if you add them to an area which needs new cells, they can differentiate into that type of cell. So in this case, you need heart muscle cells. So if you add the stem cells there, they can differentiate into heart muscle cells and repair the damage. So that's how you get 2 marks here. So first thing to say is stem cells are undifferentiated and they can turn into other cells. So this means, therefore, they can turn into heart muscle cells. And that's how you get 2 marks there. Second part of the question, the scientists could have used human embryonic stem cells to make the patches. 2 advantages of using stem cells from the person's own cells rather than embryonic stem cells. So why is it better for them to use their own cells rather than using embryonic stem cells? What's so good about using their own cells? Because it's their own, it has the same DNA as them. The cells won't be rejected by the body. So that's one point we can say. The cells will not be rejected. So what can happen is if the body thinks it's something that doesn't belong in the body, it could reject it, which means it sends an immune response to it, which isn't good. And that could happen if the cell is from somewhere else. But if it's already that person's cell, if it's got the same DNA as the cell, then the body is going to recognize it as their own. So it won't reject it. The other thing is some of the ethical issues surrounding embryonic stem cells. Some people say that using embryonic stem cells is killing a potential human life. So by not using the embryonic stem cells, you're basically not damaging an embryo. So there's no risk to damaging an embryo. You can also say there's no risk of a potential human life being destroyed as well. Okay, so here we've got a question on therapeutic cloning, which is all to do with stem cells. So what they've done here is they've given a bit of background into therapeutic cloning, and they've given how it works here. So they've said the embryo produced contains the same genetic information as the patient, and the stem cells taken from the embryo is stimulated to divide to form cells the patient needs. Then the embryo is destroyed. And basically they're asking you to evaluate that whole piece of information. So we need to give 2 advantages for part D and 2 disadvantages for part E. Part D, advantages, well, it's used to cure diseases, right? So that's a very good thing. So we can say can cure diseases. As well as this, remember, because it's used using the patient's own DNA, it's less likely to be rejected. The body won't reject these stem cells because it's not from somewhere else. Now we ask for 2 disadvantages. So the first thing you can say is the whole ethical issue that the embryo is a potential human life and you're basically destroying a potential human life. The other thing is that this isn't a simple procedure. You need to actually put the stem cells into the patient. We don't know if there are any side effects or there could be some risks to the patients as well. We don't fully know. This is quite a new technique. So you could mention that as well as the second point. And you can say other things here as well to get the marks. You can say that you could transfer viral diseases accidentally, or you can talk about the fact that it's got a very low success rate, this whole procedure. Okay, so this is a question on diffusion. Here we have 4 different cells and they're all surrounded by carbon dioxide molecules. And the question's asking, out of these 4 cells, which one will move carbon dioxide into the cell at the fastest rate? So here what you need to look at is the concentration gradient. That's the difference in concentration from the high concentration area to the low concentration area. The bigger the concentration gradient, the faster the rate of diffusion. So you're basically looking at the one which has the biggest difference in the number of molecules. And if you look at all of these, what you can see is, for B, for example, the number of molecules inside and outside are roughly the same. So it's going to have quite a small concentration gradient. So diffusion is not going to happen fast here. Same situation with cell C. Even though in total there are molecules, the difference in molecules between the inside and the outside is pretty much the same. They're both very concentrated, so it's not C either. For D, you have a high concentration in the inside and you have a low concentration on the outside. So what's going to happen is diffusion is actually going to go out of the cell here. So the answer here must be A, because there's a low concentration inside and a high concentration outside. So the particles are going to move inwards from the high to low concentration. And then your reason for this, for your second mark, you can either say it has a steeper concentration gradient, or you can say it has a bigger difference in concentration between the high and the low area. So I'm just going to write steeper concentration gradient. And that's how you get 2 marks. So let's look at this surface area to volume ratio calculation from biology paper 1. You can see here we have 2 different cubes and each cube has a different length. We have one cube here where the lengths are 1 and another one where the lengths are 2. This question is all about the second cube, cube B, and it's asking us to work out the surface area to volume ratio. And what it's done here, it's basically helped us to calculate this in steps. So the first thing the question is asking is calculate the surface area of one face of cube B. So that's just one face. So that's basically a square. You're trying to work out the area of that square. Area of a square is width times length. So we just do 2 times 2 here, and that gives us an answer of 4. Second question is using that 4 that we just got, calculate the total surface area of a cube. Well, a cube has 6 faces, and we've just calculated that one face is 4. So to work out the total surface area for the whole cube, you just times the 4 by 6, and that gives us an answer of 24. Now, we've worked out the surface area, we need to work out the volume. The formula for working out the volume of a cube is length times width times height. So, as they're all 2, we just do 2 times 2 times 2, which gives us an answer of 8. So the volume is 8. Therefore, the surface area to volume ratio is 24 to 8. Here we have a multiple choice question on diffusion. So the question asks, how does glucose concentration in blood compare to the glucose concentration in the small intestine? And just before that, it tells us that most of the glucose is absorbed by diffusion. So that's our key point here. If it's moving by diffusion, it's moving from a high concentration to a low concentration. So here it's going from the small intestine into the blood. So that must mean the small intestine has a high concentration and the blood has a low concentration. So the answer here is the concentration in the blood is lower. Right, so what we have here is a tricky 5 marker on diffusion and surface area to volume ratio. So what this question asks, first of all, we've got 2 cubes. Cube X represents a bacteria cell and cube Y represents a small multicellular organism. So we can see quite clearly cube X is much smaller than cube Y. The question then says a bacteria cell can absorb all the nutrients it needs by diffusion through its outer surface, but a multicellular organism cannot do that. And it wants us to explain why. And it wants us to use calculations. So what type of calculation do we need here? If you get cubes like this, it's usually just calculating surface area to volume ratio. So what we want to do is calculate the surface area to volume ratio of X and Y, and then we're going to compare them. So let's split up our calculation into 2. We've got cube X on this side and cube Y on this side. Surface area to volume ratio, let's start off with cube X. Let's work out the surface area of that. So remember, the area is of a square, so we want to do length times width here, and all the lengths and widths are the same. So it says here it's 0.00002. We can just change that into standard form. 1, 2, 3, 4. So that's 2 times 10 to the minus 4 times 2 times 10 to the minus 4. That's going to give you the area of 1 square here, right? So I'm just going to write that as 2 times 10 to the minus 4 squared. Let's work out what that is first for one face. So that gives 4 times 10 to the minus 8. Remember, that's only for one face. For the surface area of the whole cube, we want to times that by the number of faces in a cube, which is 6. So we're going to now do 4 times 10 to the minus 8 times by 6. And that gives 2.4 times 10 to the minus 7. So that's our surface area. Now let's work out volume. So volume is length times width times height. So we're going to get that 2 times 10 to the minus 4. And we're going to multiply it 3 times. So I'm just going to write cubed. So in our calculator, that's what we're going to write. And when you do that, you should get an answer of 8 times 10 to the minus 12. So our surface area to volume ratio of cube X is 2.4 times 10 to the minus 7 to 8 times 10 to the minus 12. Now we need to do exactly the same for cube Y. So let's start off with the surface area. Let's do exactly the same thing. We're dealing with easier numbers here. It's just 11. So we're going to do 11 squared, which is 121. We're going to times that by the number of faces in a cube, which is 6. And that gives us 726. So that's our surface area. For volume, we're going to do 11 cubed. Putting that in the calculator gives us 1,331. So our ratio for this cube Y is 726 to 1331. So now looking at these two ratios, it's not clear just by looking at the numbers which one is a bigger ratio. There's no fair comparison here. What we want to do is we want to change one of the sides of the ratios for both cube X and cube Y to the same number. Easiest one to do is 1. So we basically want to convert the ratio to the same scale. So we can either make both of the first numbers 1 or the second numbers 1. So let's say we wanted to make the second numbers 1. So that would be the 8 times 10 to the minus 12 and the 1,331. If you want to turn 8 times 10 to the minus 12 to 1, you want to divide it by itself. We need to do the same thing on the other side. We need to get 2.4 times 10 to the minus 7 and also divide that by 8 times 10 to the minus 12. If you do something to one side of a ratio, you have to do the same to the other side to keep that ratio the same, right? So let's do that. Let's do 2.4 times 10 to the minus 7 divided by 8 times 10 to the minus 12. When you put that in your calculator, you should get 30,000. So the surface area to volume ratio of cube X can also be written as 30,000 to 1. Let's do the same to this side. We're going to divide both sides by 1,331. So we're going to divide this side by 1,331. That's going to give us 1. And the other side we're also going to divide by 1,331. So 726 divided by 1,331, that gives you an answer of 0.55. Now both of these ratios are to the ratio a number to 1. So we got 30,000 to 1 and 0.55 to 1. So this is a much easier comparison. It's much easier to say now that cube X has a much bigger surface area to volume ratio than cube Y. So we have done the calculation here, but the question says to explain why does this mean? For the multicellular organism, it can't take in nutrients by diffusion. So what we need to say here is basically what we have found from our calculations. So that's the 1, each unit of volume we have. The cube X has a much larger surface area. So we need to write that in words. And then for the final mark, we need to relate this to the diffusion rate. So simply said, because they have more surface area to each unit of volume, the rate of diffusion is much faster for bacteria cells. In a multicellular organism, that diffusion rate is no way near as fast. It's 0.55 to 1. Diffusion is way too slow. So for humans, for example, we can't diffuse in oxygen directly from the air. That's why we need lungs to take oxygen into our body, and we need a whole transport system to distribute around the body because our surface area to volume ratio is not big enough. So we just need to write one final point here to just say the diffusion rate per unit volume is smaller in multicellular organisms. And there we go, that's how you get 5 marks there. Right, so here we have a 6 marker on transport in cells. This basically covers diffusion, osmosis, and active transport. So what we have here is a root hair cell, and we've got 3 different types of substances trying to enter the root hair cells, and we basically need to describe the processes by which the different particles would enter. So I think the easiest one to deal with here is water. Water always enters by osmosis. Osmosis is the movement of water particles from an area of high water concentration to a low water concentration, or you could say water potential as well. And it occurs through a partially permeable membrane. So water, we can say, enters by osmosis from an area of high water concentration, which is basically a solution, in the soil to a low water concentration in the cell. And remember, this occurs through a partially permeable membrane, so let's mention that as well. Right, now let's look at phosphate and magnesium now. Let's start off with the phosphate. So the phosphate are the shaded in circles. So what can we see here? We can clearly see there are so many more phosphate particles outside in the soil compared to inside the cell. There's only 2 in this diagram. There's only 2 inside the cell, all the others on the outside. So we have a high concentration of phosphate ions on the outside in the soil. So it's going from high concentration to low concentration. So if we think back to our definition, that's basically diffusion. So phosphate, we can say, enters by diffusion from a high concentration in the soil to a low concentration in the cell. Okay, so we talked about 2 of them now. Now let's look at magnesium ions. Let me use a different color for this one. So for magnesium, so these are the circles with the crosses in them. So what we can see, there's only about 1, 2, 3, 4, 5 magnesium ions outside in the soil. Inside, there is a much higher concentration of magnesium ions, isn't there? So this time, we're going from a low concentration to a high concentration inside the cell. So low concentration outside in the soil, high concentration inside the cell. We're going against the concentration gradient, and that's going to mean we need to use active transport, which uses energy from respiration. So magnesium enters by active transport from a low concentration in the soil to a high concentration in the cell, and we can say using energy from respiration, because this is not a passive process. The other 2 are passive processes. So maybe that's an extra point that we could put for water and phosphate. It's a passive process. For phosphates, because it's diffusion, that's also a passive process. A passive process means it doesn't use energy. There we go. All right, let's look at this question here. It's 5 marks. It says plants need nitrate ions to make proteins. A plant is growing in soil flooded with water. Explain why the plant cannot absorb enough nitrate ions. 5 marks here, so we're going to need quite a few points to explain what's going on here. So why can't the plant absorb enough nitrate ions? So we need to think in our head, how do plants usually absorb ions? This process is active transport, right? And active transport, if you remember, is not a passive process. It needs energy. Where does that energy come from? It comes from respiration. Aerobic respiration needs oxygen. So what's happening here? The plant is growing in soil flooded with water. So it's basically waterlogged. Too much water. And if you think about it, if it's growing inside water, there's not much oxygen reaching the plant, right? So if there's not enough oxygen reaching the plant, that means it's not going to have enough oxygen to respire. If it doesn't have enough oxygen to respire, it's not going to release enough energy. And if it doesn't have enough energy, it can't perform active transport. Therefore, it can't take in these nitrate ions. That's the explanation. We just need to write this down in 5 marks. So remember, you can write in bullet points. So let's start off writing how the ions are taken in. So ions are taken into plants by active transport. We can then explain what active transport is, which is the movement of particles. We can say against the concentration gradient, or you can say from an area of low concentration to an area of high concentration. Next point, we need to state that this process of active transport needs energy. Where does that energy come from? It comes from respiration, or from aerobic respiration, which requires oxygen. Right, so we've got 4 points here. Why is there not much oxygen? Because the plant is waterlogged. There's too much water. This flooded with water, it says in the question. So we can say the plant is flooded with water, so oxygen cannot reach it. And there we go. 5 marks, 5 points. That's how you get them. Right, root hair cells. Here we have 2 different scenarios with root hair cells. We've learned active transport, diffusion, and osmosis. So it's basically asking us in these 3 scenarios, which process is taking place. So what we need to look at here is what we're dealing with first of all. So remember, for diffusion is the movement of particles from an area of high concentration to an area of low concentration. Active transport is the opposite of that. It's the movement of particles from a low to high concentration. It's going against the concentration gradient. Osmosis is basically diffusion with water. So the first thing we can label here is the one with water must be osmosis, because that's diffusion with water molecules. Now, for the other two, we've got two different sets of ions. We need to look at which area has a higher concentration and which area has a lower concentration. So remember, the ions are trying to get into the root hair cell from the outside. So for the nitrate ion here, you can see there's a high concentration outside, there's a low concentration inside. So if the ions need to go inwards, then it's diffusion that's going to occur, because that's from high to low concentration. Then for magnesium ions, we have a low concentration outside and a high concentration inside. So it's going to be quite difficult for these ions to get in. They need energy because they're going against the concentration gradient. Therefore, it needs active transport. Right, so what we have here is a required practical for osmosis. So what they're using here, usually in this required practical, you use potatoes, but here we're using chicken eggs. And what we're doing is placing 5 eggs in acid for 24 hours to dissolve the eggshell. And then we're recording the mass of each one. Then we add these eggs into distilled water. So a container containing distilled water in it. That's just pure water. After about 20 minutes, we remove the eggs and we pat them dry with a paper towel and measure the mass again. So this is just like the potato experiment. Instead, we're just using chicken eggs. So first question is, a student suggested that the results for egg 4 was anomalous. Do you agree with the student? If we look at the changes in mass here, we can see we've got around 3, isn't it? So this is going from 73.5 to 77. This is around another 3. Increasing from here to here is around 3. But then if you look at number 4, it's only increasing by around 1.5. It's almost half of the other ones. Even this one is 3. So I would say it is an anomalous. So yes, we can say the change in mass is much lower than the others. Next one asks you to calculate the percentage change of mass in egg 3. That's this one here. So if you want to work out the percentage change of something, what you want to do is do the change over the initial. Change over initial and then times that by 100. So let's put the numbers we've got here. The change is between 72.4 and 75.7. So the change is going to be 75.7 minus 72.4 over the initial value. What did we start off with? We started off with 72.4. We get that, we multiply it by 100 to turn it into percentage, and the answer you should get is 4.6%. Now for part C, it's an explain question. Why did the mass of the eggs increase? So this question is all to do with osmosis, so we need to explain it in terms of osmosis. So remember, osmosis is to do with water. So what's happening if the mass of the eggs is increasing is water is moving from the water outside into the egg. And the only reason that's happening is because the water concentration outside the egg is greater than the water concentration inside the egg. So it has to pass through the partially permeable membrane of the egg, and then it makes it heavier, the mass increases. So we can say water enters by osmosis from a high concentration in the beaker to a low concentration in the egg. And then to get that third mark, mention the partially permeable membrane whenever you get a question on osmosis. Okay, so the last part is explain how the student could modify the investigation to determine the concentration of the solution inside the egg. So what they've done here is they've just used one beaker with distilled water in it. But to work out what the actual concentration is inside, we need a few more solutions. We need one with pure water, and then we need some with a different concentration of sugar in them. So you get about 5 beakers and you do the same experiment, and then you see how much the mass changes or whether the mass does even change. So something like pure water has a very high water concentration, the mass is going to increase. Then your next beaker might be 0.1 mole per dm cubed of sugar. So that's slightly lower water concentration. So the mass might still increase, but it will increase slightly less compared to the pure water. And then as you move up in concentration, the increase in mass will get less, and some of them will show a decrease in mass. because at some point your water concentration in the solution is going to be lower than the water concentration in the egg, and that's going to cause water to leave the egg. Our aim in this is to find out what that concentration is. So you'd start off with use 5 different beakers with different sugar solutions and repeat the method. So what you're doing is you're finding the change in mass, then you're finding the percentage change, just like we did in part B. And what you're going to get is loads of different concentrations with different percentage changes. So there's a graph of percentage change against concentration. And once you plot your percentage changes, this is what you're going to see. So at the very low concentrations of sugar, you're going to have a high percentage change. The mass is going to increase. And then as you decrease, it's going to get lower and eventually it's going to turn negative. What you want is the point at which it goes from positive to negative. So you'd get a ruler and you draw a line of best fit from all your points. And this point here, where the line of best fit crosses the x-axis, that is the concentration of the egg. That's how you work it out. So we just need to explain this in words for 2 more marks. So we'll say plot a graph of percentage change against concentration. That's our second mark. And then third mark, find the concentration where the line of best fit crosses the x-axis. And there we have it, 3 marks. Okay, so here we have a question where we have some students investigating the effect of different concentrations of sugar in 4 different drinks. And what they say here is the movement of water across a partially permeable membrane. So if you get given this in an exam, you should be able to identify that this is a question on osmosis, because osmosis is basically diffusion with water, and it occurs from a high water concentration to a low water concentration, and it occurs across a partially permeable membrane. So if you see that, that's what needs to be in your head. So the student made 4 bags out of these partially permeable membranes, and they put equal volumes of a 5% sugar solution in each bag. Then they weighed each bag to find their mass, and then after leaving the solution for 20 minutes, they reweighed it to see if there's a change in mass. So we can see 5% solution here, and then all of these 4 different test tubes have 4 different concentrations in them. That's going to affect the final mass of the tube. Part I asks, the bag in drink A got heavier after 20 minutes. Explain why. So let's look at tube A. So you can see here, it's 0.1% sugar solution. Whereas the sugar solution inside the tube, remember, all of them have a 5% sugar solution. So if there's a 5% sugar solution inside and 0.1 outside, it means the one outside is more dilute. There's more water in it. So the lower the concentration, the higher the water concentration. So that means inside, for tube A, there's a low water concentration. So that means the water is going to move from the area of high water concentration to the area of low water concentration through the partially permeable membrane. So this is a 3-mark question, so we need to mention all of those points to get 3 marks. So what we can say is the water concentration outside the tube is higher than inside. I said tube here, but I actually mean bag, because that's what they say in the question, isn't it? So this means water moves into the bag, and then we can say how it moves into the bag. It moves in through osmosis. So that should give you 3 marks for the first part. Second part, which drink, A, B, C, or D, would you expect the bag to show the smallest change in mass? So what you need to think of here is those factors that affect diffusion. Remember, osmosis is a type of diffusion. And we said concentration gradient affects the rate of diffusion. If your concentration gradient is smaller, diffusion happens slower. What does concentration gradient mean? Concentration gradient means the difference in concentration between the 2 areas. So remember, inside each of these bags, we have a 5% sugar solution. So if we're looking for the one with the smallest change in mass and the slowest rate of osmosis or diffusion, then we're looking at the one with the closest value to 5%. So that would be 4.2%. So because 4.2% is the closest to 5%, the difference in concentration or water concentration, because we're talking about osmosis, is very similar. So it's a very small difference. So that means the rate at which water enters the bag is very slow. So the answer here would be B. And it asks us to explain why for 2 marks. So we can either say that it's got the closest concentration to the 5%, or you can say it has the smallest concentration gradient. Closest concentration to the 5% bag. Therefore, that means osmosis is slower. So if osmosis is slower, it means less water is going to enter that bag in the same amount of time. So the final mass is going to have a smaller increase compared to bag A, for example. Here's a quick 3 marks on osmosis, the definition of it. The question asks, what is osmosis, for 3 marks. So we do need to be a bit more detailed than normal. So what we need to talk about is that it's the movement of water from an area of high water concentration to an area of low water concentration. So let's start off by writing that down. Talking about water is very important here, because remember, it is essentially diffusion with water, and it happens across a partially permeable membrane. So the movement of water from an area of high water concentration to an area of low water concentration. There's different ways you can get these marks, by the way. You might have been taught to use water potential instead of water concentration. That's fine as well. So you can say from an area of high water potential to an area of low water potential. Or you might have even learnt it as from a dilute solution to a more concentrated solution. That's also fine. So I've chosen this one. That gets you 1 mark. The other 2 marks is to actually mention the membrane that it goes across. You have to say that it's partially permeable. So we say through a partially permeable membrane. And that's 3 marks. Right, we have a question on osmosis here. We've got a method for carrying out the potato experiment. So this is when you get chunks of potatoes, you cut them up into pieces, you record their mass, then you add them to solutions for a set amount of time. So here it says 30 minutes. And then after that, you take them out, pat them dry, and record their mass again. So the question first asks, why did the student dry the surface of each potato piece with the paper towel in step 5? So when you take it out of the solution, there's going to be excess water on the potato. So you don't want that to be included in your readings. So you pat it dry to remove the excess water. Otherwise, your mass reading will be too high. So we can say to remove excess water as, we'll say increase the mass reading. Then for the second part, for part F, they've actually given you an example of results. So when they measured a potato piece at the beginning, it was 2.5 grams. Then they left it in the solution for half an hour, and half an hour later, it increased to 2.7 grams. What they want you to work out here is the percentage increase. So to work out the percentage increase for anything, or just percentage change, I always tell students it's change, whatever the change is, over the initial value times 100, right? So what they've done here in this question is they've actually made it easier for you. They've given an easier equation here. They've said increase over starting mass times 100, which is basically the same at the top. So what you do is you sub the numbers in. So the increase in mass, so what's happening is it's going from 2.5 to 2.7. So the increase in mass there, it's going up by 0.2 grams. So you do 0.2 grams, which is your increase, divided by the starting mass, not the final mass. Starting mass, it says here, is 2.5 grams. So we do 0.2 over 2.5 times by 100 to turn that into percentage. And when you put that in your calculator, you should get an answer of 8%. So here we have a question on the required practical for osmosis, the potato experiment. So here we've got a method for how to carry out the experiment already stated in the question. And it says that a person has cut 3 pieces of potato with the same length and then dried them using a paper towel, then weighed them, and then added them to different sugar solutions. Then they waited for 2 hours, took them out, dried them again with a paper towel, and re-weighed them, as well as measuring their length again. This way they could see whether their length and mass has changed. Has it increased? Has it decreased? Has it stayed the same? So we've got a diagram here, figure 1. Why did the student dry each piece of potato before weighing it? So these potatoes are going to have water on them. Especially when you take them out the solution, they're going to have water on them, and that's going to add to the mass. It's going to make the measurements less accurate when you do that. So you need to pat them dry with a paper towel to remove the excess water. So that's what you'd write, to remove excess water, as it will increase the mass. Now for part B, what 2 changes would you expect in the potato in tube A after 2 hours? So tube A, we've got pure water in there because it says water at 0 moles per dm cube. That means there's no sugar in that tube at all. It's pure water. And the potato itself, inside the potato, there is a sugar solution in there. They just naturally have sugar solutions inside them. So that means the water concentration outside in the water is higher than the water concentration inside the potato. So what's going to happen is the water from the outside enters the potato by osmosis, and that makes the potato longer and heavier. So it will increase in mass and it will increase in length. Those are the 2 that you need to tick there. Complete the sentences for part C. Water moves into and out of cells by a process called, like I said earlier, osmosis. Water would move something, the potato cells in tube A. So into or out of is the question. Like I said, high water concentration outside, low water concentration inside. So it would move into the potato in tube A. The solution outside the potato in tube A is at a something concentration than the solution inside the potato cells. So you got to be careful here. It says is at a something concentration. It doesn't say water concentration. So you have to say is at a lower or higher. So remember I said there's a high water concentration outside, which means there's a low water concentration inside the potato. Water concentration. This is talking about just concentration. So concentration of sugar. So if there's a high water concentration outside, that means there's a low sugar concentration. When they just say concentration, they mean sugar concentration, not water concentration. So you would actually say it's at a lower concentration than the solution inside the potato cell. You could also say at a more dilute concentration as well, if you wanted to get the mark. Then part D, the potato in tube B did not change. One conclusion that can be made from this observation: if the mass or the length did not change, that means no water has entered or left, or no net water has moved in or out of the cell. The only way that can happen is if the solution outside has the same concentration as the solution inside. So if we know the sugar solution outside is 0.4 mole per dm cubed, it means the inside of the potato is also 0.4 mole per dm cubed, because there's no change in concentration, no change in water concentration, so there's no net movement of water. So the conclusion would be the potato cells have a concentration of 0.4 mole per dm cubed. There we go. How does water move into and out of cells? That's what this exam question is asking for 1 mark. The answer here is osmosis. The key here is that it talks about water, and diffusion of water is basically osmosis. That's how water moves in and out. It's not breathing. That's how air gets in and out of the body. And respiration, remember, that's the transfer of energy from glucose. Next question. We've got 3 different cells here. Cell P, Q, and R. And it asks, into which cell, P, Q, or R, will water move the fastest? So 2 keywords here, fastest and into. Which of these will the water move into? You need to look at the percentage of sugar solution here. So what you need to remember here is the higher the sugar solution, the lower the amount of water. The more water there is, the more dilute the solution is, therefore the higher the water concentration. So which of these has the highest water concentration and which of these has the lowest water concentration? The 1% one would have the highest water concentration. The 3% one would have the lowest water concentration because it's got more sugar in it, so it's less space for water. It only has 97%, whereas the 1% one has 99% water. The 2% one is still high. It's higher than the cell R because that's got 98% water. So what's going to happen is the water is going to move into cell R. You might get some water also going from the 2% to the 1%, but the question's asking into which cell will it move the fastest. That's going to be cell R because it's got the lowest water potential. So the answer here is R.